So after this S phase, the next phase under interphase, which is the last phase, is what we call the nuclear membrane disintegrate. 2. Condensation of the chromatid. We have 3 major things we'll be talking about under cell cycle. Hello everyone, welcome to Arithmetic Challenge. Today we'll be talking about a specific topic which most people feel it is actually very simple but yet complicated. What am I talking about? We are talking about cell cycle. Although we'll be focusing on mitosis, we want to describe the cell cycle in its entirety before we nail it down to talk about the mitosis. Alright. So when you are talking about cell cycle, you are referring to the cyclical event that occur within a cell from the point when that particular cell is produced or is made to the point where it will give rise to two identical daughter cells. Just like human beings that reproduce and give birth to new offspring, the same thing applies to every cell of your body. So when you are talking about cell cycle, I want your mind to just think about reproduction. It's a way by which the cell tries to reproduce and carry out their own entire life. Just like human beings, they give birth to a human being and then at the end of the day carry out a lot of activities, and then at the end of the day the person or individual will die. The same thing applies to this point too. But before the cell dies, they make sure they also reproduce. So every activity that takes place from the period of one cell division to the beginning of another, that's what we call the cell cycle. And this cell cycle has 3 major phases. The first one is the interphase. The second one is what we refer to as the M phase, that is the mitotic phase, and the last part is the cytokinesis. Each of these 3 parts has a role to play. The first part, which we call the interphase, is illustrated with the diagram here. This interphase has 3 major stages: the G1 phase, the S phase, and the G2 phase. That is why I use this as an annotated diagram to explain and illustrate what we are talking about. I said the interphase stage comprises of 3 major phases: G1 phase, S phase, G2 phase. But before I nail these things down one by one, there is something you need to understand first. Why exactly do you think cell cycle has to take place? Why exactly do you think cell division in general has to take place? One of the main reasons for this is because in some cases you might notice that, okay, for individuals who exercise a lot, there may be some point in time where you will have issue with your tissue, whether tearing, injury, and what have you. And it is through this process of cell division that these cells are repaired and worn out tissue are being repaired to make sure your body can perform optimally. So if there is no cell division, if cell cycle is not present, automatically we should understand that it will be very hard for new cells to be produced. Just like a similar example is when the right kidney is cut off, automatically the left kidney also will hypertrophy. What causes that hypertrophy of the left kidney is due to the presence, or let me say, this complete cell division that is taking place within that particular left kidney. And as a result of that, it will make it to be able to perform the function of 2 kidneys. So, moving back to interphase now. Having known the main function of our cell cycle, which is to repair worn out tissue for normal growth and development, these phases will be talking about them one by one. The interphase is usually regarded as the first phase and also the resting stage. In most textbooks that you might come across, you will notice that they don't usually include these with any process of mitosis or any process of meiosis, because everybody considers the interphase as a resting stage. You know, that particular point in time when you plan to travel, and then the process by which you are packing your bag, that is what we can consider as the interphase stage. That particular way or that particular time that you are beginning to pack your load to make sure that you do not forget anything, that stage can be categorized as a synonym to interphase. So, nailing it down now, when you're talking about the interphase stage, I say the first stage is G1 phase. For the G1 phase, it is the longest phase. As you can see within the diagram here, this phase is actually bigger compared to the other phases I have here. The G1 phase is the longest phase of any interphase stage, and it is during this stage that the cell is still working actively and functioning optimally in preparation for cell division that is coming. So at this stage now, the cell is still resting, performing its function. Let's take the liver cell as an example now. When the liver cells are in the G1 phase now, automatically one of the things that will happen is that they will perform their function. One example of the function of a liver cell hepatocyte is to detoxify. So at that particular stage, you can say that liver cell, which is the hepatocyte, is still detoxifying in its normal ability. But it will get to a particular point in time before it enters S phase. There is something we call the G0 phase. Let me write it here, the G0 phase. Some period of time may come where the cell in particular may not want to enter the next phase, which is the S phase. It will enter a phase we call the G0 phase. This G0 phase is a phase where the cell decides that at this point in time, I'm not going to undergo any cell division. There are some certain things that might cause this. One of which is in a situation where such cell lacks adequate amount of nutrients, oxygen, and whatever for normal growth and development of such cell, it will not undergo normal cell division. Although there are some certain cells in the body that does not undergo cell division. Example is the nerve cell. The nerve cell, when they reach their normal size, they do not undergo cell division. And such cell, we can say they are in permanent D0 phase. And some other, like let's say the hepatocyte again, if the liver cell is damaged now, it may enter the D0 phase, waiting to get the required nutrients till it will now be pushed to the S phase. So this phase is the phase where the cell is not deciding or is not willing to enter the next phase as a result of the fact that there is inadequate of some certain nutrients or there is a damage to the particular cell. And in some cases, let's now assume that this cell is not able to easily move to the next phase for a long period of time. What will happen is that such cell will die off. That's what we call apoptosis. Apoptosis. So such cell will die off, so that what this means is that the cell is not useful anymore. And to prevent any kind of problem for the body, the best thing to do is to kill that particular cell. And that's what this apoptosis actually works on. So let's now imagine that a particular cell entered the G0 phase, and then at the end of the day, the adequate nutrients and whatever it means to enter the S phase is there. So the next thing it will go to is the S phase now. The SS is represent synthesis, the synthesis phase. At this phase now, I want you to have something at the back of your mind, especially when you are talking about mitosis. During the S phase, there is a replication of DNA of the chromosome. There is a statement I made earlier. When it comes to mitosis, you have to understand that new daughter cells will be formed. New daughter cells are formed. So that means a particular cell like this, let's say cell A will later produce cell B and cell C. B and cell C. So the process by which it produces two different cells is what we refer to as mitosis now. So the process of producing two different cells from a mother cell is what we call mitosis. And for this to take place now, this daughter cell has to look exactly like the mother A. So automatically, it is during this S phase A that the DNA of the mother will be replicated, such that each of the daughter cells that will later be formed will have the same identity as that of the mother. The S phase will just be the phase where there will be replication of DNA to make sure that each daughter cell at the end of the day, they will have their own clear identity, which will have the same genetic constituent as that of the mother. And because of that now, each cell can now function independently on their own. So after this S phase, the next phase under interphase, which is the last phase, is what we call the G2 phase. The G2 phase. I said something earlier when I was talking about G1 phase. I said it is the phase where, if you want to think about it, just categorize it as a phase where, imagine when you are traveling now, and then you try to pack your luggage to make sure that you do not forget anything. This is where the what we are talking about. But for the G2 phase, this is the stage where you start rechecking your luggage to make sure you do not leave anything behind. The S phase is where you put in all your documents, required documents, to make sure that everything is intact. But the G2 phase is where you start to check again to make sure that everything is intact and then you are ready to travel. So what do I mean by this now? The G2 phase is the phase where the cell will try to cross-check, is there anything that we need to add? Is there anything that we need to remove? So as to make sure we are ready for mitosis, which is the M phase. So at this particular stage, there are some proteins are produced so as to make sure these mitosis that will take place after will occur without any issue. So that's what these two phases is actually about. And then for the mitotic phase now, the M phase, which is where we are actually going, entering the mitotic phase proper now. So all these other phases can be regarded as the resting phase. That is, we are preparing to enter the mitotic phase, which is the mitosis aspect. So automatically, the next thing we are going to be talking about now is mitosis properly now. And the first stage of mitosis is what we call the prophase. Prophase is regarded as the first stage of mitosis. For you to understand this thing better, there are some certain things you need to take note of importantly. There are four major things that occur during this prophase stage. There are four major things that occur during the prophase stage. The number one thing there is that there will be condensation of the chromatid. There will be condensation of the chromatid. What do I mean by this now? When we were in interphase stage, I want you to understand something. During the interphase stage, the chromatid are not that visible. They are very tiny. That is under the microscope. But at this phase now, they condense. They condense together and they become more visible to the extent that they are joined together at this particular point, A, which we call the centromere. The centromere is the point that joins two sister chromatid together. The point that joins two sister chromatid. And remember, we said this sister chromatid, there's duplication of their DNA during the S phase. So after they are duplicated and then they enter the G2 phase, that is how we are able to get to the prophase stage. So these two sister chromatids are attached at the centromere, and then it makes this chromatid become more visible through condensation. That's the first thing that happens during prophase. The second thing that happens during prophase is that the nuclear membrane begins to disintegrate. The nuclear membrane is the membrane that covers the nucleus, the inner part of the cell. As you can see with the diagram I have here, it has already started disintegrating, such that the nuclear membrane will not be there anymore. So the chromatid will just be within the cytoplasm directly like that. That's the second thing that happens during the stage of prophase. Remember, I want you to have something at the back of your mind which I will be repeating. The fact that mitosis is taking place to make sure we have two daughter cells which are identical to the parent. So here now we have chromatid, and I said the nuclear membrane will begin to disintegrate. The third thing that will happen is that centrioles and mitotic spindle begins to form. Centrioles and mitotic spindle begins to form. These centrioles will be the part where this mitotic spindle will attach to. The main reason we need this part of this cell is because it is within this spindle that the chromatid will later align on, and then they will later separate. When we get to that aspect, we are going to get what I mean. But at this point now, I want you to understand that the centrioles and then the mitotic spindle are formed during the stage of prophase. And for the fourth thing that happened during the prophase stage, remember I said four things are going to happen. One, nuclear membrane disintegrates. Second, chromatid become more visible. Third, centrioles and mitotic spindle are formed. For the fourth part now, the centrioles migrate to the opposite side of the cell. The centrioles migrate to opposite side of the cell. Just imagine me taking this aspect like this and then turning it this way, so that one end of the centriole is air, the other end is air, so that it will now be one on either side. So those are the four things that happen during the stage of prophase. One, nuclear membrane disintegrates. Two, condensation of the chromatid. 3. Formation of centrioles and mitotic spindle. And 4. Migration of the centrioles to the opposite pole of the cell. So the second phase we are talking about now is metaphase. This metaphase comes after the prophase stage. Remember, during the prophase stage, I said there are 4 major things that happens there. So immediately after 4 of those events has taken place, we are going to enter the phase where we can consider it to be the metaphase stage. During the metaphase stage, this is how the cell is going to look like. At the metaphase stage now, there are some certain things peculiar to this stage. As you can see, there is a difference between this diagram and the diagram for the prophase. Majorly because there is alignment of a chromatid on the center of the cell. That is, they align on the spindle fiber, the mitotic spindle. They align on the mitotic spindle. They arrange themselves in the center of the cell, and which is quite different from what happened during the prophase. During the prophase, the cells are not like this. They are just enclosed or within a nuclear membrane which is already trying to break down. But at this stage, the nuclear membrane has already disintegrated, and as a result of that, they move towards the middle of the cell, aligning themselves on what we call the mitotic spindle. So the mitotic spindle will now be the one to house them during the metaphase stage. And at this stage, another thing that also happens is that, or let me say, you should understand, is the fact that these chromatids are still attached together at the centromere. Remember, during the stage of prophase also, we said sister chromatids are joined together by what we call centromere. During this phase too, the centromere still join the 2 sister chromatids together. So this is what metaphase entails. So the third stage we're talking about is anaphase. Anaphase stage is a continuation of what happens at metaphase. So everything that happened at metaphase, the continuation is what we are going to be talking about during this stage now. During the stage of metaphase, remember I said the chromatid, they align on the center of the cell, specifically on the spindle fiber, the mitotic spindle. During anaphase stage now, the centromere that joins the 2 sister chromatids together has already separated. So as a result of that now, each sister chromatid will move to opposite pole of the cell. Remember, the main aim of mitosis is that we want to try to produce 2 new daughter cells. So that means at the end of the day, we are going to have a daughter cell which will have its own genetic constituent, and another daughter cell which will have its own genetic constituent at the same time. So during the stage of anaphase, what this does this with is separation of the centromere such that the sister chromatid will now be able to move away. During the stage of anaphase also, we can consider this chromatid as chromosome. Now we call them chromosome during this anaphase stage. The last stage we're talking about is telophase. The telophase stage is just a continuation of the anaphase stage. During the anaphase stage now, I made mention of some certain things that happen. Everything that happens there, the continuation is what will happen under telophase now. One of the things that you should take note of under telophase is that the chromosomes decondenses and it becomes less visible. It becomes less visible. That's the number one thing that happens. And also the nuclear membrane that has already disintegrated during the prophase stage begin to reform. It begins to reform back around each of these genetic constituents. This is because at the end of the day, we want to produce 2 new identical daughter cells, which are identical to the parent cell. So automatically, each of them will have the same identity as the parent cell. And also, the third thing that happened during the telophase stage is that it is totally ready for the process of cytokinesis. It is ready for the process of cytokinesis. So cytokinesis is the last thing we are going to be talking about now. Remember when I started this video, I made mention of the fact that there are 3 major things we'll be considering under cell cycle. 1 is the interphase stage, 2 is the M phase, and then the third thing is the cytokinesis. And we've been able to successfully explain interphase and M phase now. So for cytokinesis, we are talking about the division of the cytoplasm so as to make sure each cell have their own organelle to function independently. Within the cytoplasm of any cell, there are a lot of organelles there, talking about mitochondria, talking about reticular reticulum, and each of them have their own specific function. To make sure this cell is able to perform independently, the cytoplasm that connects them has to degenerate. And this is what cytokinesis is all about. As you can see with the diagram we have here, the cytoplasm begins to divide into two, such that at the end of the day, we are going to have two different cells having their own nuclear membrane. And within this nuclear membrane is where we have the chromosome, which consists the genetic constituent. At the end of the day, as you can see, one cell is splitting into two. A singular cell is splitting into two. As a result of this, we can say cell division has occurred, thereby making sure that there is replication of cell for normal growth and development of the body. And just like I gave you the main function or the main reason why cell cycle has to occur, the same way is how this same process is what it will entail to carry out these activities. So in conclusion, cell division just deals with a cycle of events that occur within a particular cell, from the point when it is produced to the point when it will also produce another cell. That is the cyclical events that occur, within a cell. That's what we refer to as cell cycle. And I said we have three major things under it, which is the interphase, the M-phase, and the cytokinesis. Interphase stage is considered as the resting stage. M-phase is where the cell division proper takes place. Cytokinesis is where the cell divides into two equal parts so that each cell can function independently. If you have any question, kindly drop them in the comment section and I am going to reply to everything one by one. And please, lastly, don't forget to subscribe, like and share. I will appreciate this. Thank you very much.